Hello students, this is Dr. Amarpreet Kour, a lecturer of Jammu and Kashmir Education Department. Today I am going to start with sexual reproduction in higher plants, that is in angiosperms. Before starting the chapter, I have request to all of you. Kindly keep the textbook of biology of class 12th handy, okay? And this book is by NCERT. I always refer the standard book. It is certainly of NCERT. And open the book at page number 20. There is a figure 2.1, and it is labeled as a diagrammatic representation of LS. That means longitudinal section of a flower. Because the first thing that we are going to study in this chapter is flower. Okay? A flower is a gametophytic part of angiosperms, and it is responsible for sexual reproduction in them. A typical angiospermic flower has four parts. The first one is known as calyx. In the diagram that I have mentioned at figure 2.1, you can see the green sepals. Okay, the technical name of sepals is calyx, and they are sterile part. They don't take part in sexual reproduction directly. So these are not very essential parts of the plant. Second, you can see the yellow colored petals in the flower, and they're in the technical name for these yellow petals is known as corolla. Okay, corolla means petals. They are also sterile part. They are not essential for flower in reproduction, but their function is to attract insects for pollination. Later on, I will discuss what is pollination. In the same diagram, you can see a bunch of stamens. The technical name of stamens is androecium. Is a male part, okay, and fertile. Certainly, it is fertile when it is a male part. Then in the center, you can see a different structure, and it is labeled as a stigma. Okay, stigma or a hole, you can say it is a carpel. Carpel is the female part, and stigma is present at the tip of carpel. The technical name of carpels, in this diagram you have a single carpel, but in many flowers there are many carpels. So you can say a carpel or carpels. The technical name for them is gynoecium, and certainly it is fertile because it is a female part. Now I will discuss two things in very detail. One is the androecium or stamens, and other gynoecium or carpels. All of you kindly go to the figure 2.2 where where it is written a typical stamen. It is a figure A, and 2.2 figure B. There is a three-dimensional cut section of an anther. So kindly go to figure 2.2. I'm starting with stamen, that is also known as microsporangium. You have studied in bryophytes and pteridophytes, and a typical stamen, it consists of two parts. Clearly, it is seen in the picture. It is made up of a filament and anther. The terminal bilobed structure, that is greenish color, and there is a red strip in these two bilobed structure. This is the anther, and the long tube-like structure is known as filament. The important part in stamen is only anther. Okay. However, there is functional part of filament too, but from sexual point of view, anther is the most important as it contains the pollen grains, or it is the microsporangia. A typical anther is bilobed, and two lobes are separated by a deep groove in them, and these two lobes are attached with each other with a central sterile tissue. Okay, this is the sterile tissue and it is known as connective. Between the two lobes there is connective, which is a sterile tissue, and two lobes, they are fertile. If you cut an anther in a transversely, you will find four lobes. Okay, these are actually four microsporangia and they are located at the corners of each lobe. Hence, a mature anther is a tetrasporogenate. That means four sporangia are present in an anther. Microsporangia, it forms the pollen sac, which on maturity is filled with pollen grains. Now, structure of microsporangium. In the figure, you can clearly see figure 2.2. A typical microsporangium, it consists meristematic tissues, that is the primary sporogenous cells, and it is surrounded by anther wall. Primary sporogenous cells forms microspore mother cells, and these cells later on form pollen grains. Firstly, anther wall layers. It is made up of four layers: epidermis, endothecium, middle layer, and tapetum. I'm starting from the outside. Okay, beginning from the outside. Firstly, it is epidermis. The next layer is endothecium. The third layer is middle layers, and the innermost, it is the tapetum. Now the epidermis. It is the outermost, single layered and protective in function. It has no other function than protection. Now endothecium. Cells of this layer have alpha cellulosic fibrous bands, and these bands are responsible for dehiscence of anther due to their hygroscopic nature. Hygroscopic means ability to attract water and become swollen. Due to this character, it helps for the bursting of anther wall and releasing of pollen grains only when they are mature. Now, middle layer. Cells of this layer are ephemeral. The cells of middle layer, they are firstly three layered, okay, and these cells are ephemeral. That means they are for short period. They degenerate at maturity. Now the innermost layer, this is known as tapetum. This is the innermost wall of anther layer, which is surrounded by sporangious tissues. These cells provide nourishment to developing grains. They have the same function that have you seen in animal reproduction, the serotoli cells. Okay, they also provide nourishment to sperms. Now, sporangious tissues. When the anther is young, a group of compactly arranged homogeneous cells are called as sporangious tissues. Okay, they are present at the center of each microsporangium, and their function is only to synthesize pollen grains, and the process is known as microsporangiosis. This is the process which helps in the formation of pollen grains. I will discuss it in detail. Firstly, at the four corners of anther, there are present microsporangious tissues, okay, at the four corners. And These undergo haploid meiotic division and they form microspores which are haploid. Okay, and they are present at the four corners of this tetrad. This whole process, like from microsporangineous tissues, okay, to pollen grains, is known as microsporogenesis. As the anther matures and it dehydrates, the microspores they dissociate from each other and develop into pollen grains. Inside each microsporangium, there are thousands of microspore, or you can say pollen grains, and they are released with dehiscence of anther. Now, pollen grains. The pollen grains are the male gametophyte, and generally they are rounded in shape, and they have the diameter of 25 to 50 micrometers. The cell wall of pollen grains is known as sporoderm, and it is made up of further two layers, axine and intine. The axine, it is the outer layer and it is made up of a typical component known as sporolinin. It is one of the most resistant organic material known. Kindly note down. It is the most resistant organic material known. It can withstand high temperature, strong acids, and strong alkalis. It also helps in fossilizations. That means to form the fossils. Because this layer is resistant to all the extreme conditions, so they can survive from years to years and they can form fossils. There is an aperture, okay, in this axine wall. This aperture is known as germ pores. At this particular place, sporolinin is absent. Now, intine. It is the inner layer and it is made up of cellulose and pectin. And the third part is known as cytoplasma. Cytoplasma of pollen grains, it is surrounded by a plasma membrane. A pollen grain or a microspore, it divides mitotically and it forms two cells. One is vegetative cell and other one is generative cell. Vegetative cell is bigger in size and it reserves the food material. The generative cells, it is spindle shaped and it has abundant cytoplasma and nucleus. Remember, only generative cell it divides to form two male gametes, and pollen is shed in 3-cell stage. Clear? Vegetative cell it reserves the food material. Generative cell it divides into two male gametes, and at this particular stage, at 3-cell stage, the pollen grains are shed. Thank you for listening to my lecture. In case you have missed any of lectures, you can visit my website, that is www.amarpreetkour.com, and you can download the lectures from there, and also you can post any comment or query.